Hi there, in this video we are going to discuss about the biosynthesis of the collagen. Biosynthesis of the collagen. Previously we have discussed about the structure of collagen. So next we start with the biosynthesis of collagen. So the collagen molecule is synthesized basically in the fibroblasts. Fibroblasts are the cells which lead down the collagen at the site of damage of tissues, and they lead to the formation of collagen at scar tissue and lead to the scarring. They are also synthesized by chondroblasts which form cartilages, and they are also synthesized by osteoblasts which form the bones. So depending upon the tissue type, they are synthesized by different precursor cells. So what happens, we previously discussed that collagen is synthesized from the alpha chains which are being coded by alpha 1 and alpha 2 genes. So these genes are transcribed into the messenger RNA, and this specific messenger RNA for collagen has a specific sequence here, or you can call it signal sequence, which lead to the formation of then this messenger RNA lead to the formation of proteins. It goes to the ribosomes, and that ribosomes move to the rough endoplasmic reticulum and start synthesizing the polypeptides of the collagen. Then the polypeptides also have the signal sequence at their end terminal, which directs these polypeptides into the lumen of the rough endoplasmic reticulum. So what has happened till now? Uh, the alpha 1 and alpha 2 genes are transcribed, and they lead to the formation of messenger RNA. Messenger RNA goes to the ribosome bind to the rough endoplasmic reticulum, and there is synthesis of polypeptide chain which is directed into the lumen of the rough endoplasmic reticulum. Okay, so the newly synthesized polypeptide precursor of the alpha chain are called newly synthesized polypeptide precursor of alpha chain are called pre-pro alpha chains. Pre-pro alpha chains because they have special amino acid sequence at their end terminal ends. They have special amino acid sequence at their end terminal ends, and this sequence is a signal that targets the polypeptides chains into the rough endoplasmic reticulum, from where they go to Golgi, and then they are packed for excretion out of the cells. And when they go into the rough endoplasmic reticulum, this signal sequence is cleaved. From them. Now, after this cleavage, the peptide chain are called pro alpha chains. So initially, as a result of translation, the chains formed are called pre-pro alpha chain, which have sequence at their end terminal, which targets them to the rough endoplasmic reticulum. Inside the rough endoplasmic reticulum, they are cleaved and form pro alpha chains. Okay, so what happens? These pro alpha chains, which are known into the lumen of rough endoplasmic reticulum, they have we previously discussed the primary structure of collagen like glycine, X and Y. They have at Y position either proline or lysine. This proline or lysine are hydroxylated. The hydroxyl group is attached to them to form hydroxyproline, hydroxyproline and hydroxylysine. This hydroxylation reaction occurs in the presence of vitamin C, iron, and oxygen O2. How this reaction occurs? The enzyme with catalyzing this reaction is prolyl hydroxylase and lysyl hydroxylase. So the chain formed pro alpha chain, which have at the Y position either proline or lysine, is hydroxylated to hydroxyproline and hydroxylysine. This reaction occurs in the presence of vitamin C, molecular oxygen, and ferrous, and this reaction is catalyzed by either prolyl hydroxylase or lysyl hydroxylase. How this reaction occurs? Let me show you. Supposedly, this is the prolyl residue here. Then here comes the molecule of alpha ketoglutarate, which gets converted to succinate. Alpha ketoglutarate gets converted to succinate plus a molecule of carbon dioxide, and here comes the oxygen, which gets converted to water, enzyme catalyzing reaction is prolyl hydroxylase, and here comes hydroxyprolyl residue. So prolyl residue comes here. Alpha ketoglutarate reacts and gets converted to succinate plus carbon dioxide. Oxygen comes and gets converted to water. Enzyme catalyzing the reaction is prolyl hydroxylase, and hydroxyproline residue is formed, and this reaction occurs in presence of iron and vitamin C. So this hydroxyproline and hydroxylysine, they are having hydroxyl group or OH group. This OH group enhances the interchain hydrogen bonding. It enhances the interchain hydrogen bonding, which increases the strength of collagen. So if there is deficiency of vitamin C, when vitamin C is deficient, then this hydroxylation reaction is impaired. When this reaction is impaired, then the hydrogen bonding is impaired. When hydrogen bonding is impaired, the strength of collagen is impaired. So when strength of collagen is impaired, there is decreased strength of the connective tissues. That's why the condition occurring due to deficiency of vitamin C is called scurvy. Scurvy, in which there is the weakened connective tissue of the body due to weakened collagen, and the patient of scurvy frequently have bruises on the skin because their capillaries are weak and fragile, due to which blood leaves the capillaries due to rupture of capillaries, and there is extravasation of the blood due to capillary fragility due to deficiency of vitamin C occurring in the scurvy. Okay, so till now we have discussed that the pre-pro-alpha chains were formed, which had signal sequence at the end terminal, which directed them into the rough endoplasmic reticulum. Inside the rough endoplasmic reticulum, they were cleaved to form pro-alpha chain, and then in pro-alpha chain there is hydroxylation of proline and lysine residue. Next step is glycosylation. The hydroxylysine residue are glycosylated here. Glucose or galactose molecule are attached to them, which further enhance the interchain stability of the collagen. So what happens after hydroxylation and glycosylation? Pro-alpha chains form pro-collagen. Then the pro-alpha chains become arranging themselves, and when they arrange, they form pro-collagen. 3 chains unite and form pro-collagen. And in this pro-collagen, what happens? This central region has the peptide chain formation. However, at the carboxyl terminal, there is the formation of the disulfide bonds. Here is the formation of disulfide linkages. These disulfide linkages occur between chain as well as within chain. So at C terminal, this is the formation of disulfide bond. Similar structure also exists at the N terminal. So in the center there is triple helical structure of collagen, and at C terminal this is propeptide, and at N terminal this is propeptide present. Then what happens? Then this structure goes to the Golgi apparatus, and from Golgi apparatus it is released outside the cell. It is released outside the cell than in extracellular matrix. Then in extracellular matrix, N and C procollagen peptidases work. N and C procollagen peptidases come, and these N and C procollagen peptidases cleave this structure here. They cleave this structure, and then the collagen molecule is freed into the extracellular matrix. So what has happened? Hydroxylation of pro-alpha chain occurred, then glycosylation of pro-alpha chain occurred, then the central portion of there arranged into a trihelical structure, while at C terminal there was disulfide bond formation. This is called propeptides here, and there is propeptide here. Then N terminal, then it was packed into the vesicles, and these vesicles by Golgi apparatus were released outside the cell, and outside the cell these C and N terminal extra peptides were cleaved, and when they were cleaved, a collagen molecule was released here, and this cleavage reaction occurred by N and C procollagen peptidases. So this structure formed is called tropocollagen molecule. It is called tropocollagen molecule. Then these tropocollagen molecule arrange themselves in the form of fibrils. Tropocollagen molecule form the fibrils by overlapping. For example, now this is a tropocollagen molecule. Another tropocollagen molecule comes here and overlaps it. Another tropocollagen molecule comes here and overlaps it. Then here comes the tropocollagen molecule and overlaps. Here comes the tropocollagen molecule and overlaps. In this way, these topocollagen molecules overlap each other and join together by hydrogen bonding and so on. So they form the long fibrils of collagen. They form the fibrils of the collagen, and these this is overlapping is occurring by 3/4 of the molecule. So they overlap, topocollagen molecule overlap and form the collagen fibrils. Then here comes another enzyme which is called lysyl oxidase. Here comes an enzyme called lysyl oxidase. What this enzyme do? The lysyl oxidase enzyme, what does it do? This enzyme basically uh, converts the lysine into L-lysine residue. Okay, so it removes the amino group or NH2 group of the lysine and converts it into the C double bond oxygen H alkyl group or aldehyde group. So let me elaborate structurally here. Supposedly this was the chain going on here, and here is the, if I draw the lysine structure, and CH2, here is also CH2 and CH2, and here is NH2. This is supposedly a lysine residue of the collagen here. So what does do lysyl oxidase? It removes this NH2 and converts this structure, this bondage, into the alkyl group or aldehyde group, C double bond oxygen and single bond with H. So if this structure created by lysyl oxidase enzyme, which is called al-lysine or al-lysyl residue, if it is come near another amino group here, which can be due to second amino acid. Let me elaborate. Supposedly this was a chain, this was the lysine residue here. Lysine residue normally has NH2 here and then CH2. Lysyl oxidase removes this NH2 and converts this CH2 into the bond like alkyl group H. So if this alkyl group comes near with another NH2 group or NH2 containing amino acid, then what will happen? They will react and they will form a bond here. They will form a CNH bond, and this will be from other chain. This is second chain and this is first chain. So in this way, this fibril will link each other and there will be further cross linking, forming the mature collagen molecule. Let me repeat it. The pre-procollagen was directed into the rapidoplasmic reticulum, from where it formed procollagen. Procollagen had the pro-alpha chain had the hydroxylation and glycosylation, and then they joined together from center to form procollagen molecule, which had propeptide at C and N terminal. It was released, then C and N terminal regions were removed and tropocollagen molecules were formed. Tropocollagen molecules arrange themselves in the form of fibril. Then comes an enzyme lysyl oxidase, which removes the NH2 group from certain lysine molecules. When an H2 group from certain lysine molecules is removed, it gets converted into the alkyl group. This alkyl group, if it comes near the NH2 group from some other chain, then it reacts with it and it forms bond with it, then there is again further cross linking and it further gives strength to the collagen molecule. So in this way, the collagen is matured and mature collagen molecules are formed in the cells. Next, we should be knowing some diseases due to collagen, abnormal synthesis of collagen, which are called collagenopathies. Collagenopathies, and these collagenopathies include Ehlers-Danlos syndrome, and osteogenesis imperfecta. What happens in Ehlers-Danlos syndrome? In Ehlers-Danlos syndrome, it is a heterogeneous disorder, heterogeneous generalized connective tissue disorder. What happens in it? The fibril collagen molecule is not formed properly. The fibril collagen molecules are not formed properly due to enzyme defect. Which may be defect in lysyl hydroxylase or pro-collagen peptidases. Or there can be mutation in the amino acid sequence of collagen type 1, type 3 or type 5. Okay, so typically it involves defect in the type 3 collagen. Typically, it involves defect in the type 3 collagen. Okay, so it is a disorder, heterogeneous disorder of connective tissues due to defective synthesis of collagen. This defective synthesis can be due to defect of lysyl hydroxylase enzyme, pro-collagen peptidase enzyme, or due to mutation in the genes synthesizing collagen type 1, type 3, or type 5. Typically, it is due to defect in the type 3 collagen. So, collagen mutant chains are synthesized which are not normal, and when they are not normal, what happens? There is abnormal blood vessels. There are abnormal blood vessels due to which cardiovascular complication can arise, and moreover, the skin changes occur due to which skin is hyperelastic. The skin is hyperelastic and stretchy skin, and the joints are also loose due to these collagen defects, loose joints. Other disease which occur due to defective collagen synthesis is osteogenesis imperfecta, and this disease significantly affect bones as well. So, it is also a heterogeneous disorder in which bones bend easily and fracture easily. Bone bend easily as well as fracture easily, and there is also the twisted spine leading to the humped back in this disease, or kyphotic appearance, humped back, kyphotic appearance due to spinal changes, and it has two types: osteogenesis imperfecta, tarda, uh, and this disease is due to decreased production of alpha 1 and alpha 2 collagen chains. When these chains are less than normal, then the collagen synthesis will also be less than normal. So, it can be characterized by early infancy fractures. For example, in the intrauterine life, there can be fractures of the bones of the infant, and there is type 2 osteogenesis imperfecta. Type 2 osteogenesis imperfecta is more severe form. In this condition, there is also a pulmonary hypoplasia in utero, and it can lead to the death of the fetus. And the most patient with osteogenesis imperfecta have the defect in the genes of the chains for the polypeptide chains. Uh, so, what happens in these mutation? Usually, the glycine is replaced by other amino acids, and this glycine replacement lead to the abnormal structure of the collagen. So, osteogenesis imperfecta is another disease due to defective collagen synthesis. It usually affects the bones bend and fracture easily. There is spinal changes due to which humped or kyphotic appearance occur. It has two types: osteogenesis imperfecta tarda and osteogenesis imperfecta congenita. So, both lead to the skeletal changes. In osteogenesis imperfecta congenita, which is more severe, pulmonary hypoplasia also occur and intrauterine death is reported. So, typical mutation here is the replacement of glycine with other amino acid with bulky side chains. Okay, so it was about diseases. So, how collagen is degraded? Because connective tissue changes continue to occur, and connective tissue is a dynamic structure. So, collagen are degraded by enzymes called collagenases, and these collagenases are a part of tissue metalloproteinases. These are basically enzymes which are present in the extracellular matrix, and they modify the structure of extracellular matrix from time to time. So, they cleave the collagen at the specific sites as well as into their constituted amino acids. So, degradation of collagen occur from time to time by specific collagenases enzyme, which are the part of tissue metalloproteinases. So, it was all about collagen synthesis and collagenopathies and degradation of collagen. Thank you.